Of DNA. Today we're going to look at two different techniques, PCR and gel electrophoresis. We're going to look at the amplification, or how we make more copies of DNA using polymerase chain reaction, and then we're going to look at the use of gel electrophoresis to sort these DNA fragments out. In a video a little bit later on, where they're going to join these two things together to look at how we can interpret gel runs to help us with DNA profiling. Finally, we're going to finish up looking at our final technique, which is called polymerase chain reaction, or PCR, which amplifies fragments or makes lots of copies of them. Okay, so applying traces of DNA. So often when we're at a crime scene, for example, we're only able to extract small amounts of DNA, which is not enough for a process like gel electrophoresis. So using PCR, it allows DNA to be amplified through the use of that initial piece of DNA that we've found. This is how we can get loads of DNA from saliva sample or a blood sample that's found at a crime scene. There's a really great YouTube clip here which shows you this process, so please have a look at that. We need to know the stages of PCR and the details around them. So PCR is made of 3 stages: denaturing, annealing, and extension or elongation. This diagram here shows you everything that you need to know about the steps in PCR. So we need to be able to recall these steps. We need to memorize these. Because this is something that's Googleable, the chances of it being asked on a SAC is minimal, but it's very, very common that you'll be asked on an exam to recite these steps. And in reciting them, we need to know what happens at each of those stages and also what temperatures they occur at. So the first thing that happens in this process of PCR is that our DNA is denatured. So the DNA sequence is denatured, so it dissociates or separates into 2 strands. This happens at 94 degrees for approximately 2 minutes. Okay, so step 1 here, we've denatured the DNA, causing it to separate, 95 degrees, approx. The second thing that happens is that we bind primers. So short segments of DNA strand, known as primers, are added. These primers pair with regions of the DNA, with regions at either end of the DNA of interest. This happens at 55 degrees. Okay, so we've added in our primers and the primers have annealed to the DNA. They've joined to the DNA. The third thing that happens is that these primers extend or elongate. So the polymerase enzyme uses these primers as a starting point and extends them or adds base pairs so that we end up with 2 complete double strands of DNA. For this step, we need to supply nucleotides. So we need to have lots of ATs and Cs and Gs in our PCR machine, and it happens at 72 degrees. Okay, so we've added our primers, which provide a starting point for the replication of our DNA. We've got nucleotides, and those primers are then extended. Okay, so at 72 degrees, those bases are added so that we end up with complete strands of DNA at the end of this process. Here's a question to sum up a little bit of what we just discussed with respect to PCR. So we've got radioactively labeled nucleotides were incubated with an unlabeled molecule of DNA. Appropriate enzymes were added to the DNA and the DNA was allowed to replicate for 2 cycles. Examine the following information or the following key. So we've got unlabeled DNA. We've got 1 strand of the unlabeled DNA, which has got that little asterisk, which indicates our radioactive nucleotides, and then we've got both strands here that have got radioactive nucleotides. So given that 2 cycles of DNA replication has occurred, the end result would be, so in our first cycle, we would have one of our original strands plus one of our new strands added. So both of them would look like that, one new and one added. In our next cycle, we're going to go from 2 strands to 4 strands. So we'd end up with one of our originals, one of our new, another one of our originals, another one of our new, because they all end up being separated. And then we will have a new strand added to each of those using our radioactive nucleotides. Okay, so in our solution, we're looking for a picture that sort of reflects this process. So where answer wouldn't be A because they've all got original strands. We only end up with 2 original strands at the end. Okay. It wouldn't be D because that shows us that we haven't separated the DNA in the first place. It wouldn't be B because there's no template strands present there. So it would be C that represents the end of that second cycle. Electrophoresis, as I said before, helps to separate DNA based on their lengths. So the longer lengths of DNA move slower than the faster ones. So this particular technique involves placing DNA into a gel that's connected to an electrical charge or an electrical current. It's important for us to note that DNA has an overall negative charge, and this is due to the phosphate in its sugar phosphate backbone. So it's drawn down to the positive pole of the gel. So this is our DNA electrophoresis machine. Okay, it's got a power source, and our negative electrode is positioned at the top here, and our positive electrode is at the bottom. DNA is loaded into these little wells here at the top. So this is where our DNA goes. We pipette it using a special pipette into those wells. And when we turn on our power source, because the DNA is negatively charged, its molecule is negatively charged, it will move down our gel towards the positive electrode at the bottom of our electrophoresis machine. So the larger pieces of DNA, they will get stuck at the top, and the smaller segments of DNA, they'll move faster and further down the gel. The reason that the larger ones get stuck or end up staying towards the top is because the gel is porous, so it's got little pores in it. So because the large fragments of DNA are too big to fit through those pores, whereas the smaller ones are able to. So they'll move down faster, which allows us to be able to order these fragments based on their size. So as I said, the fragments have to weave their way through the gel matrix, and the longer ones remain at the top because they get stuck, and the smaller ones move down the bottom. There's a link down the bottom here to a really good gel electrophoresis demonstration, so I'd encourage you to please watch that as it shows a really good animation of this happening. Here we see some DNA fragments that are getting loaded into the gel and the electric field drawing them down. So let's have a little bit of a closer look at this gel. So our gel electrophoresis, the gel inside of it is an agarose gel with an electric field. So pores for fragments to move from negative charge down towards the positive charge. It contains a buffer solution, and we've got a blue dye that we add to make it visible, and we use UV or extra dye to look at the bands after it's run through the gel. After a gel has been run and we've dyed it blue and we've put it under UV light, this is the result that we get. So we can see that the negative terminal is up the top here, so the DNA would have been loaded into these wells at the top. Just going to change my pen color. So the DNA would have been loaded up here. It's traveled down towards the positive electrode, and these little bands indicate DNA fragments. So bigger ones will be at the top, smaller ones will have traveled further down the bottom. You might be thinking, how do we know how big these fragments are? Because it's all well and good to have them sized from smallest to largest, but a lot of the time we need to know exactly what size those fragments are for us to be able to identify them. So to interpret gels, which is another thing that's outlined as part of your study design, a DNA sample with known fragment size is added into one of the wells, and it's allowed to run. So those fragments then separate out, and because we know exactly how big they are when we put them into that gel, because there are known markers, so this sort of, I suppose, acts as a little bit of control. It's our point of comparison. It allows us to be able to work out the fragment size of an unknown piece of DNA. So for example, if we have a look at our estimated fragment sizes, all we have to do in our unknown source is compare them to the markers of known source. So we've got a fragment here which sits between 6,682 base pairs and 4,361. So this particular piece of DNA here, we would estimate is probably going to be roughly, I would say, 4,000 base pairs in length. We've got another one here. So it's between 4,316 and 2,322. Okay, it's about in the middle. So we're going to say it's approximately 3,000 base pairs long. We've got another one just here and another one just here. So this bottom one down here, it's very close to that 564 range. So we're going to say it's approximately 580 base pairs long. Then we've got this guy up here, who's a little bit further down from the 2000. So we might say that this one here is approximately 1,500 base pairs in length. So we're using the known markers to help us to estimate the fragment size of the unknowns. Gels are also used to examine DNA evidence, such as DNA found in crime scenes or paternity disputes. A region of a gene is collected and has a restriction enzyme applied to it. Okay, so we've cut up a piece of DNA, and sometimes we may add a probe to it, and a probe is just a piece of DNA of known length and of known genetic sequence that provides us with a little bit of a comparison too. We're going to look at gel electrophoresis now. So we've already looked at restriction enzymes as being our scissors that we use to cut the DNA into different pieces. We've also looked at the enzyme ligase and said that this is our glue that we use to glue pieces of DNA together. We're now going to look at gel electrophoresis, which allows us to be able to sort out fragments based on their size. So this allows us to determine the allele an individual has. So people with the big C would only get a band at 1165 base pairs, because that's how long that particular copy of DNA for that allele is. A person with big C, little c, they would show a band that's this length here, the 1165, but they would also have a band which is 879 because they've got both of those copies of those alleles in their genome. A person with little c would show only bands at 789. So we can use this knowledge to help us to determine the paternity of a child, or if someone's DNA has been found at a crime. This here, we've got a gel. It's got person T, person J, and person K. Okay, and it's got in this particular lane 4 some DNA of known length or known size. So we can use this here to determine paternity and also whether people have been located at a crime scene based on what banding they show. So here's a little question. Who is the child of Mary's baby? Why? Look at the band of the child that reflects the allele, and all alleles have to be received from a parent. So Mary is the mum, and Bob and Larry are possible dads. She doesn't know who it is. So in order for us to be able to work that out, we need to compare the DNA that the child has. So the child has a matching band with the mum, which makes sense because we already know that she's the mum. Half of the child's genetic information will be coming from her. Now we have to look at the other pieces of DNA and see what we can match. Okay, so child has a common piece of DNA to Larry. And then if we go down to the next banding of DNA, all three of them have got the same for that particular allele. So we wouldn't be able to use that to help us in this particular case because we're looking for points of difference to try and work out who the dad is. But the child here has also got another matching piece of DNA fragment size or allele that's been inherited from Larry. So in this particular situation, we'd say that Larry is the child's father. Another question, during a fight between a number of people, one was seriously injured. Blood samples were taken from the victim and the crime scene and 4 suspects. DNA was extracted from white blood cells in each of the blood samples and electrophoresis of a sample was carried out. The results are shown in the diagram below. The question is asking us the person most likely to have been at the crime scene is suspect 1, 2, 3, or 4. So what we need to do here is match up samples that the victim has and samples of the suspects. So the victim has got these particular DNA markers. Now we have to look for matches between what's at the crime scene and whether they match each of our suspects. So if we look at suspect 1 has no match here, no match here, indicating that they weren't at the crime scene. Suspect 2, there is one matching piece there, one matching piece there, and one matching piece there. So it's looking very much like suspect 2 is the one that was at the crime scene. Suspect 3, there is also one matching band at the top. Okay, there's no matching band here. And this one is slightly further below that one, so there's no matching band here. Okay, suspect 4 has no matching band, no matching band, no matching band, so definitely not suspect 4. And given that suspect 3 only has that one matching band, it's probably just a coincidence. It might be a common allele. We would say that suspect 2 is most likely, because that's what our question asks us to be at the crime scene. Using gel electrophoresis, scientists discovered that there were 4 types of blood on Otzi's clothing. The results were as follows. So this is another question that's come from a past VCAR exam. And our question B, part I, is asking us which sample of Otzi's clothes actually belongs to Otzi, which blood sample. Okay, so here we've got blood sample taken from his blood vessels. Okay, blood sample 1 from his clothes, blood sample 2 from his clothes, 3 and 4. Again, what we're doing here is just looking to match the bands to see which clothes belong to Otzi. Okay, so we've got a matching band here for blood sample 1 and 2. We've got a matching band here for sample 1 and 3. This one doesn't match, either does that one. Okay, we've got a matching band here, and we've got bands here that don't match to the blood that's been found in Otzi's vessels. Okay, so the fact that in blood sample 3 there's 2 bands that have been found, it's most likely that the blood sample 3 clothes belong to Otzi. Propose one hypothesis that would explain the presence of other blood samples on Otzi's clothes. Okay, so for this particular question, we're just going to skip to the next to show us that blood can come from other people or other animals. That's why it can be located on the clothing.